Hello, everyone. Thank you for attending the live webinar, Understanding the Potential Impact of the Gut Microbiome on SARS-CoV-2 Research. Today's speaker is Dr. Heesun Shin, Product Manager in the Clinical Next Generation Sequencing Division at Thermo Fisher Scientific. During the webinar, if you have any questions, please use the chat box in the presentation window, and we will answer as many questions as we can after the presentation. If we cannot get to them, we will follow up with you after the live event. Now I would like to introduce our speaker today, Dr. Heesun Shin. Heesun Shin focuses on developing and managing NGS-based products for human health research. Her most recent launch was the ION AmpliSeq Microbiome Health Research Assay. This, along with the recent release of the Complete ION Reporter Microbiome Analysis Solution, allows for an optimized microbiome study. Heesun, you may now begin your presentation.
Great, thank you for the introduction. It's great to be here today. In today's presentation, I would like to discuss the potential impact of the gut microbiome on SARS-CoV-2 research. And this is because emerging research shows that there may be an association between the clinical manifestation of SARS-CoV-2 infection and the gut microbiome. And it's been well documented that a reduction in the diversity of the gut microbiome, which is known as dysbiosis, is associated with a number of diseases, including respiratory infections. So one of the big questions surrounding SARS-CoV-2 infection is, why is there such a big disparity in the severity of the disease among individuals? And then the ultimate question is, how can we develop effective treatments and vaccines to help combat this disease? So as SARS-CoV-2 continues to impact billions of lives across the globe, exploring a potential association between SARS-CoV-2 and the gut microbiome is an important consideration, which may lead to the development of more effective intervention strategies in the future. So with that said, I'll move on to the agenda for today's presentation, which will be in 3 parts. So in the first part, I'll give a little background on the gut microbiome and how imbalances in the system can have a significant consequences for a number of diseases, including infectious disease. And in the second part, I'll introduce potential implications for the gut microbiome in the context of SARS-CoV-2 infection. It's specifically related to gut microbiome and lung axis, gut and lung axis, and implications for disease severity and potential role in vaccine efficacy, as well as emerging avenues for the future research. Finally, I'll briefly introduce ION AmpliSeq Microbiome Health Research Kit, which we feel can be a key resource to facilitate research into this emerging area of study. And after the presentation, as mentioned, we'll have a few minutes for me to answer any questions you may have. Okay. So let's begin with an introduction to the gut microbiome and its emerging impact on human health and disease. So microbiome is a diverse collection of microbes that can be found naturally occurring in different locations of our body, such as the mouth, the skin, and the gut. And these microbiomes, especially those in the gut, play a major role in some human diseases. Interactions between the gut microbiome and the human host are complex and are also bidirectional. Microbes are provided with a protective habitat and nourishment from the host, and these microbes in turn are beneficial to the host by helping regulate various physiological functions, including dietary digestion, and also providing protective immunity against pathogens. The gut microbiome is also known to significantly regulate the development and functioning of the innate and adaptive immune systems. So there is this complex back and forth going on between the gut microbiome and the host immune homeostasis. So specifically, dysbiosis of the gut microbiome is associated with various immune-related diseases, such as diabetes and autoimmune diseases, as well as gastrointestinal disorders, such as IBD and IBS. Additionally, the gut microbiome can impact how well people with cancer respond to treatments such as anti-PD-L1 immunotherapy, and this has been shown in several cancer types, including melanoma, lung, and kidney cancer. More recently, it's been also documented that the gut microbiome has profound impact on the host response to infectious diseases, such as in respiratory infections like tuberculosis and pneumonia. So there is a growing need for understanding how the gut microbiome influences the immune system, and this knowledge then may help guide the development of therapeutic interventions for a growing number of important human health conditions, including infectious disease. So how is it that disruption to the diversity of the gut microbiome can lead to acute and chronic diseases? Well, it turns out that changes in the gut microbiome can have both local and systemic biological effects. So for example, some local effects that are well understood include metabolizing non-digestible nutrients, providing protection against gut infections, helping to maintain intestinal barrier functions, and also educating the immune system. However, it's being increasingly appreciated that in addition to local effects, the gut microbiome also has systemic effects through the production of anti-inflammatory metabolites, such as short-chain fatty acids, that are transported around the body, which are products of bacterial fermentation of non-digestible nutrients like fiber. In the case of dysbiosis, these important protective effects that are provided by the gut microbiome are compromised, and this contributes to the development of immune-related diseases. And another area of active investigation has been around cancer immunotherapy research, as I mentioned earlier. The recent evidence has demonstrated that dysbiosis associated with how well an individual with cancer will respond to immune checkpoint inhibitors. So in this situation, a healthy immune system is required for a person to have a good response to the treatment. And given that dysbiosis can negatively impact the immune system, it's not surprising that dysbiosis can impact how a person will respond to immunotherapy. So the current working hypothesis is that a diverse microbiome results in a diverse immune system that is better able to understand cancers or recognize cancers after immune checkpoint inhibition. A diverse microbiome and distinct microbial composition signatures are therefore potential biomarkers for how well a person is likely to respond to immunotherapy. So this gives us a sense of how the gut microbiome can have a significant impact on chronic diseases. But the gut microbiome also has a significant impact in infectious disease. So the traditional concept has been that an infection occurs in a linear progression. A pathogen enters the host. The pathogen evades the host defenses, colonizing the host and resulting in the disease. So the research has been mostly focused on understanding the mechanisms by which a pathogen can overcome host defenses and establish infection. However, more recently, research has shifted focus to host-pathogen interactions and how the indigenous microbiome is an additional element that plays a critical role in determining susceptibility to pathogens and the outcome of the infection. So for example, the microbiome plays a key role in colonization resistance as part of the mucosal immune system. which provides a physical and chemical barrier in the gut epithelium, is responsible for secretion of antimicrobial peptides and produces targeted antibodies. And short-chain fatty acids produced by certain bacteria can systematically alter the production of antimicrobial peptides and reduce the fitness of pathogens by inhibiting their growth. Alternatively, changes to the gut microbiome can result in host immune changes, leading to chronic low-grade inflammation, which can result in a greater risk for infection. And this relationship between microbiome structure and altered immune response is exemplified in the lung, where enrichment of anaerobes such as Prevotella and Veillonella is associated with an elevated inflammatory state, leading to increased infection susceptibility. So it's becoming clear that infectious diseases should not be considered a singular microorganism problem. And as research shifts to investigating the complex dynamics between microbial communities and the host immune system, we'll gain a better understanding of how infectious diseases can be prevented and how therapeutic approaches can potentially be adapted or developed. Okay, so that gives a brief overview of how the gut microbiome is known to impact human health and disease. So now let's consider how this may relate to SARS-CoV-2 research. So we have discussed that the ecosystem of the gut microbiome can both regulate and be regulated by invading pathogens, including viruses, facilitating an effective immune response. Therefore, the question arises around whether the interaction between the gut microbiome and SARS-CoV-2 may play a role in the intensity of the infection and clinical outcomes. So over the next few slides, I'd like to review 3 potential areas where the gut microbiome may have an impact on SARS-CoV-2 research, namely gut-lung axis, the gut microbiome and its association with disease severity, and SARS-CoV-2 vaccine development research. So starting with the gut-lung axis, recent research has revealed that there is a vital crosstalk between the gut microbiome and the lungs known as the gut-lung axis. The gut-lung axis allows for the passage of molecules such as endotoxins, microbial metabolites, cytokines, and hormones through the bloodstream, connecting the 2 distant organs. So for example, alterations in microbial species of the gut and the metabolites that they produce have been linked to the development of lung-related diseases such as asthma and COPD, as well as infectious diseases. So for example, in people with tuberculosis, significant shifts in the relative abundance of short-chain fatty acid producing bacteria like Roseveria, Faecalitarium, and Eubacterium have been observed. Additionally, specific microbial composition changes have been linked to influenza virus infections, and a protective role for the gut microbiome during pneumonia has also been demonstrated. So taken together, these observations suggest that immune responses at distant mucosal sites can be regulated by gut microbiome, affecting outcomes in response to bacterial and viral infections in the lungs. And interestingly, the gut-lung axis is bidirectional, and the integrity of the gut microbiome could also be disturbed by SARS-CoV-2, causing gut dysbiosis in the host, as with other infectious diseases. So these observations point towards the possible involvement of the gut-lung axis for disease progression in people with SARS-CoV-2 infections. So with respect to the gut-lung axis, some potential connections can be drawn based on a number of characteristics of SARS-CoV-2 infections. So for example, SARS-CoV-2 primarily causes lung infection through binding of ACE2 receptors that are present on alveolar epithelial cells. And ACE2 receptors are reported to be expressed in intestinal epithelial cells in the gastrointestinal tract as well. And also, intestinal epithelial cells are known to harbor SARS coronavirus, which caused the major SARS infection in 2002 and 2003. And SARS-CoV-2 RNA has also been detected in the stool of some individuals with SARS-CoV-2 infection. Gastrointestinal symptoms, including diarrhea, are also frequently observed in these individuals. And recent research indicates that SARS-CoV-2 may also be spread by fecal-oral transmission. Also, the high SARS-CoV-2 mortality and morbidity is in the elderly and in those with underlying health problems such as diabetes. And these groups tend to have less diverse gut microbiome. So given these observations, further research into the GI tract in the context of SARS-CoV-2 infection progression should be considered to gain a better understanding of the complexities surrounding this disease. So now I'm going to discuss the potential relationship between the gut microbiome and the disease severity. So during the initial SARS-CoV-2 outbreak in China, early research coming out of the first affiliated hospital indicated that bacteria in the gut may have a significant impact on the range and the severity of symptoms associated with SARS-CoV-2 infections. So after profiling the gut microbiome of the SARS-CoV-2 infected individuals, these researchers found that many of those with more severe disease exhibited gut microbiome dysbiosis. So specifically, there was a decrease in probiotic organisms, including lactobacillus and bifidobacterium, and both of these probiotics are known to have anti-inflammatory properties. And furthermore, these researchers suggest that the use of probiotics may even play a role in control of possible secondary infections and proliferation of pathogenic microorganisms. And additional evidence for link between the gut microbiome and SARS-CoV-2 comes from a study that reported over 60% of individuals with SARS-CoV-2 infection displayed gastrointestinal symptoms such as diarrhea, nausea, and vomiting. And this group of subjects also tended to have a more serious or critical form of the disease. Based on an analysis of the data, the authors went on to describe 20 proteins associated with disease severity, including immune factors known to be elevated during systemic inflammation. They then generated proteomic risk score, or PRS, based on these proteins. And using machine learning algorithms, they were able to correlate the PRS to the microbiome from study subjects' fecal samples, demonstrating an association between gut microbiome composition and disease severity. So taken together, these initial findings suggest a potential role for the gut microbiome in the pathogenesis of SARS-CoV-2 infection. Finally, I'd like to briefly discuss how the gut microbiome can potentially impact vaccine efficacy. The vaccine efficacy has a number of known confounding factors, such as pre-existing chronic infections, gut-associated pathologies, use of immunosuppressive therapies and antibiotic treatments, among others. And over the past decade, various studies have revealed that a number of these factors are influenced by the gut microbiome. So for instance, the diversity of the gut microbiome plays an important role in the production of adjuvants that play key functions in antibody production, and therefore the potential for vaccination success against infection. So together, these observations suggest that to understand how our immune systems respond to viral vaccines and to optimally modulate this response, we need to study not only the genetics of the host and the virus in relation to the vaccine, but also the relationship with the gut microbiome. And to look at this in a bit more detail, a recent research demonstrated how gut dysbiosis resulting from the use of antibiotics impacted the immune response to vaccines, using influenza as an example. So this study showed that a decrease in microbiome diversity could be observed up to 180 days after antibiotic treatment prior to vaccination. And this dysbiosis led to the upregulation of inflammation, impaired production of antibodies, and resulted in an overall dampening of the immune response. So essentially, what's been found is simple. You need to have healthy immune system to successfully produce antibodies to the vaccine, and so you're able to develop immunity against the virus. And again, the diverse and well-balanced microbiome promotes a healthy and diverse immune system. Okay, so to sum up, more research is certainly needed to get a better understanding of the association between SARS-CoV-2 infection and the gut microbiome, and the potential implications of this for optimizing therapeutic approaches for the disease. As we're all aware, there is an urgent need for effective treatments based on antivirals and vaccines. And so it's important to explore any potential intervention strategies that may exist. And one approach would be by studying the gut microbiome profile of individuals and developing effective dietary or clinical interventions to improve gut dysbiosis and thereby potentially improving overall immune response in such subjects. The prebiotics or probiotics have been shown to be effective in some studies to regulate dysbiosis. However, there remains insufficient evidence to support the blind use of conventional probiotics as a supplement. So until much more is known about the pathogenesis of SARS-CoV-2 and its association with the gut microbiome, scientists suggest that a more targeted therapeutic approach for modulation of the gut microbiome is likely needed. And finally, the role of the lung microbiome should also be investigated. Although technically more challenging to study, insights from such studies may well add a new dimension to the understanding of infectious diseases. So to conclude, in the fight against infections like coronavirus, a healthy gut microbiome is potentially a key factor in maintaining an optimal immune system, which is especially important given that both an overreactive and underreactive immune response can lead to adverse effects that could result in clinical complications. So earlier this year, we launched our new Ion AmpliSeq Microbiome Health Research Kit. We worked with our key research collaborators to develop our commensal gut bacterial species panel that are associated with immune-related disorders. As an example, I want to briefly show you a pilot study with one of the researchers to demonstrate how we can study gut microbiome as it relates to disease such as SARS-CoV-2 infection. So the data that I'm presenting here is from a researcher studying diabetes at a regional research institute that is affiliated with a local hospital so that they have access to samples from individuals with diabetes. They aim to investigate the gut microbiome composition in subjects with different types of diabetes, as well as healthy controls to better understand the role of the gut microbiome in diabetes. So in this slide, I'm showing an example data of type 2 diabetes when the authors compared samples from healthy individuals to subjects with type 2 diabetes. They saw a clear trend of increased level of Bacteroides to Firmicutes ratio. And while these are from the small sample numbers, the result is consistent with what's been published in the literature. And at the species level, an increased level of Bacteroides vulgaris and decreased level of Fecalitarium pratiniti in subjects with type 2 diabetes were observed, which is again consistent with the literature. So finally, with high accuracy and reproducibility that this assay provides, researchers are planning to investigate the microbiome profile changes over a time course to study the impact of clinical or dietary interventions as well. So one of the key features of this assay is that it's a fully validated sample to answer workflow with an analysis solution also included. The ion reporter analysis solution for this assay provides visualization options for single and multi-sample analyses, including interactive full taxonomy classification plot, stacked bar plots for relative abundance, heat map and PCOA plots for unsupervised clustering analysis, and linked to QIIME2 software, it provides alpha and beta diversity plots and matrix. And another point that I want to mention here is that the solution uses up-to-date and most widely used reference databases such as Silva database. So this assay offers two panels, one comprehensive 16S panel using 8 of 9 hypervariable regions. And this allows the global microbiome composition analysis for overall picture of the microbiome. And the target species panel for highly specific species level identification of 73 key bacterial species known to be associated with those immune-related diseases. And I want to mention here that the target species panel contains a large number of the known bacterial species with an antiviral or immune modulating properties as listed on this slide. So this assay has great potential utility for studying the association between the gut microbiome, SARS-CoV-2, and our immune system. And we look forward to hearing more about the research in this area. Okay, so with that, I'll end my presentation. Thank you for listening. And now I'll be happy to take any questions you may have.
Thank you very much, Heesun, for a very informative presentation. Let's go ahead and take a few questions from the audience. So your first question here says, can you elaborate a bit more about the significance of the fecal transmission route of SARS-CoV-2 that you mentioned?
Yeah, sure. Thank you for the question. So in addition to transmission via droplets and close contact that we are aware, fecal oral transmission has also been shown to be important in some cases. So gastrointestinal connection with SARS-CoV-2 infection that I've addressed in this presentation, and the fact that the virus has been detected from fecal samples all support the fecal oral route in the disease transmission. But the significance of this is still unclear, and so we need more research on this subject. But the possibility of the virus transmission via sewage, wastewater, or contaminated water, I guess, cannot be underestimated.
Great, thank you. Okay, our next question says, you said that it is more challenging to investigate lung microbiome. Why is it more challenging?
Yeah, a great question. So microbiomes at other sites other than the gut such as lungs, received less attention. That's because of relatively a low microbiome biomass compared to gut microbiome, and also some technical difficulties. So the studies demonstrating the presence of microorganisms in the lung changed the conventional belief that lungs are sterile, but still the progress on the lung-specific microbiome has been slow, and that's mostly due to various sampling challenges. So, for example, the respiratory tract is anatomically much more complex than the GI tract, and the microbial flow in the lungs can also be bidirectional with airflow, while it's one directional flow for gut microbiome. And obtaining fecal samples is easy and non-invasive, but sampling lungs for microbial assessment is prone to contamination, and it's difficult because of its complicated structure, and the proper sampling technique can be quite invasive. And also with limited nutrition compared to the gut, there is higher turnover rate and fewer microbiota in the lung, as I've mentioned as well. So it's challenging. However, I think it's still a very valuable area of research, as I addressed in the presentation.
Thank you, Heesun. We have time for just a couple more questions. Now let's go with this one. Is there a connection between the severity of COVID-19? Do you see the microbiome being more central to stratification of people who are at high risk?
Yeah, it's a very good point. Yes, I think it would be possible, and it is in fact actively being investigated. So it's also been shown in cancer, for example. The researchers are finding microbial signatures associated with specific cancers, and also response to therapies like anti-PD-L1 immunotherapy that I mentioned earlier. So understandably, there is interest in developing a stool-based diagnostic for risk of mortality from SARS-CoV-2 infection, and also immune-supporting microbiome therapeutics. Great question.
Thank you. Okay, and let's go ahead and take this last question for today. There are reports that antibodies will only last for a couple of months in those infected by SARS-CoV-2. Is there any evidence that the gut microbiome may play a role here?
Oh, that's also a great question. While I can't really comment on the specifics of this topic, we have been monitoring all the publications that pertains to the key areas of research. And yes, there have been recent studies or preprints suggesting that antibodies do peak and then decline after a couple of months. But clearly more work is needed to fully understand the longevity of the immune response to the virus, and also if or how the microbiome plays a role there. I'll also mention that there is still a wealth of information that is out there, and as a quick plug, you can always read more about the latest research on Thermo Fisher website, which is called Behind the Bench blog.
Thank you again, Heesun. Do you have any final comments for our audience before we go today?
Yeah, I just want to say thank you to everyone for attending this webinar on a very interesting topic. And I look forward to hearing more questions from you. Thank you.
Thank you. And I'd also like to thank the audience for joining us today and for their interesting questions. Any questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank Heesun for her time today and for her important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. You can view this webinar on demand. LabRoots will alert you via email when it's available for replay. That's all for now. Until next time, goodbye.